So the difference between a gene and a genome. Please look at this black image also when I speak, because that's when you will understand. Okay. So gene, if you break down the DNA, this is the entire, this helical structure, this is the entire DNA. Okay. Haploid structure is entire DNA. Break down the DNA, you will find genes. You can see here they have mentioned genes in this image. Okay. This is this, you can, if you see the arrow moving, this is one gene. This is second gene, third gene, different combinations. You can see how the CG-TA combinations differ. Okay, every gene has a different kind of combination of all these proteins. Okay, so it goes on and on, it forms the DNA. Okay, so it's a functional piece of DNA. Okay, pieces of genes come together and they form a DNA. Okay, and what is genome? If you look here, the DNA will wrap up into a chromosome. Chromosomes will come together and sit in the nucleus. So this entire set of chromosome, okay, how many chromosomes does human beings have? How many chromosomes does a human being have? How many pair? We have 23 pair, so total 46. Okay. Yeah. 23 pairs, so total is 46 chromosomes is what a human body has. Okay, so this different types of chromosomes is found in a nucleus. Okay, so the entire set of your chromosomes is called genome. Every human has a slightly different genome. Okay, based on their own characteristics, what they have inherited from their parents. Okay, so what is the how size? If you compare the size, only a few 100 to 1,000 base pairs will be there in a gene. Okay, but the size of a genome, if you see, so many genes will come together, form DNA. All these DNAs will form chromosomes. So the totality of the genome, okay, the size of the genome is much bigger as compared to gene. Okay, gene is just a part of a DNA. DNA is part of the chromosome. So all the 23 pairs of chromosomes will come together and form the genome. Okay. So the sequence of all these pairings, it is non-repetitive sequence. Okay, there is always a unique gene for everything. Okay, but when it comes to genome, repetitive sequences are there. What you find, a typical kind of sequence, what you find in chromosome number 1, you can find it in chromosome 19, 18 as well. Okay, so repetition is there. Location, you can find the genes on the chromosome, because chromosome is made up of DNA. Okay, and gene is a functional unit of DNA, so you can find the genes on the chromosome. And where do you find the genome? Genome is in the entire nucleus of the cell. Okay, the study of gene is called genetics, the study of genome is called genomics. Is it clear to all? This is just your introduction to genetics. Okay. Okay. So next, coming to DNA and RNA. Okay, a single strand of this haploid structure. Okay, that is your RNA, and the other when there are double helix structure. Okay, that becomes your DNA. Okay, that's the difference between RNA and DNA. So the base pairs, you can see these base pair were the ones that used to make the gene. Okay, cytosine, guanine, adenine, and thymine. You must have seen the short form as CGTA coming. You can see here CGTA if it is visible to you. Okay, so these are the ones that make sequences. Okay, these are the ones. So cytosine, guanine, adenine. Okay, these are the nitrogenous bases. Why nitrogenous bases? You require protein for this. Okay, you require protein for this. Protein provides nitrogen for your DNA repair, etc. Okay, so they are the nitrogenous bases. They come in different sequence, okay, and form different kind of genes. Okay, so this is the difference between DNA and RNA. Full form deoxyribonucleic acid, RNA ribonucleic acid. Double helical structure is what we call as DNA. If it is a single strand, it is RNA. Okay. So what are the haploid and diploid cells? So haploid cells in one set of chromosomes n equals to 23. Okay. Diploid cells, you can see here the chromosomes which has both these strands. Okay. So two sets of chromosome is there. In total sets of chromosome humans have is 46. Okay. So if you just take one part of the, in some genetic conditions like trisomy 21. Okay, can anyone say what is the other name of trisomy 21? A very common genetic condition. It's Down syndrome. Okay, so in trisomy 21, what is happening? A single homologous chromosome is extra. Okay, usually you should just have 23, but there is a the 21st chromosome. Okay, the 21st chromosome has one extra haploid set of chromosomes. So that's why it becomes trisomy 21. Tri means 3, somy means chromosome, and 21 means from 1 to 23, from the 1 to 23 pairs of chromosome which you have, the 21st pair of chromosome has tri chromosomes. Okay. So that's why it leads to a genetic condition called Down syndrome, also called as trisomy 21. Okay. So this is the difference between haploid and diploid cells. So next, coming to what is nutrigenetics and nutrigenomics. When you listen to the explanation for the very first time, it will be very confusing for you. But over and over, when you read these statements again and again, okay, you will understand where is the difference. Okay, nutrigenetics and nutrigenomics. Okay, nutrigenetics, the first one on the top. Your genes affect how your body responds to food. Your genes affects how your body responds to food. For example, there are around 45 participants in this Zoom meeting. Okay. If I give all of you a glass of cow's milk, okay, I give you all of you a glass of cow's milk, will your body digest it in the very same way? A very simple example. A glass of cow's milk to all 45 of you. And why it will not digest it in the same way? Why will not? Why will? Yeah, because of the various metabolic activities. Some are 45 of you, as we know the statistics. 80%, 70 to 80% of the Indian population is lactose intolerant, but we are not aware of it. Okay, so definitely I'm giving cow's milk. Why specifically cow's milk? Because cow's milk is a very well known allergen. Okay, when it comes to food sensitivity. Okay, so definitely all 45 of you will not digest or metabolize this glass of cow's milk the same way, because every single one of you have a different genome. Okay, you all of you have a different genetic structure. How your genetic structure is going to metabolize a glass of cow's milk is going to be different. So that is nutrigenetics. Okay, that is nutrigenetics. Now coming to nutrigenomics. What you eat affects how your genes behave. What you eat affects how your genes behave. Okay, the example, for example, a person A is on a ketogenic diet. Okay, person B is on a vegan diet. Ketogenic diet and vegan diet. Okay, completely different kind of food structure. In ketogenic diet, it is a non-vegetarian and fat-rich food. Vegan diet is naturally non. Am I audible to all? Am I audible to all? Okay. So let's take two examples. Okay. A person is on a ketogenic diet. Person A is on ketogenic diet. Person B is on a vegan diet. Okay. Why vegan and ketogenic? In ketogenic, it is fat rich. Okay. Only 5% of energy comes from carbohydrates. Remaining should come from protein and fat. Vegan diet is naturally low on fat. Okay. Naturally, when you see it is low on fat. Okay. So over a period of time, okay, they both of them were healthy individuals. They didn't had much of deficiencies or anything, but when you put them on these diets, you follow them over a period of time, okay, will they acquire certain deficiencies? If they do not consult a nutritionist, etc., they just blindly follow what is given online, etc., over a period of time, will some diseases catch up with them, their mental health issues, their diseases? Definitely, right? Because what you eat is going to affect your genes. Okay, ketogenic diet for a pretty long time, we still do not know what will be its effect. Okay, we do not know what is the long-term effect of a ketogenic diet. Definitely, your mental health will be under stress. Okay, because too much of ketone bodies in your body will definitely cause too much ketone bodies in your circulation will definitely cause some genetic mutation. Okay, the food that you eat can cause genetic mutation in you. The food that you eat can cause genetic mutation in you. Okay, so that will lead to some cancer like colon cancers, etc. People who have a very ill dietary habit, okay, very pretty bad and ill dietary habit, they are at the risk of getting colon cancer. And what is cancer? Some kind of mutation is taking place in your genes, okay, because of which you get cancer. Okay, so yeah, definitely your diet is related to your diet can affect your genes. Okay, that is nutrigenomics, how food, something external, affects something internal. That is nutrigenomics. Okay, what is nutrigenetics? Something that you already have, your internal, how it responds to external. That is nutrigenetics. Okay, is both the definitions clear to you, nutrigenetics and nutrigenomics? Okay. Next, coming to principles of nutrigenomics. So gene expression can occur directly or indirectly. Usually what happens, whatever food you eat, it definitely undergoes metabolism. When the food is undergoing metabolism, there is going to be byproducts. Okay, any chemical reaction you take, there will be some byproducts. Let's call these byproducts as metabolites. Okay, metabolites. After metabolism, whatever remains, okay, they become the metabolites. Okay, these metabolites, especially of vitamin A, D, fatty acids, zinc, etc., they have higher tendency to cause certain gene expression, good or bad. Okay, so directly or indirectly, metabolites can cause some changes in your gene expression. Okay, then your diet is a major risk factor for certain diseases. For example, you inherently know that you are born into a family who has pretty much every generation has a history of cardiovascular diseases. Okay, so your diet plays a major role in deciding what your future health will be. Okay, so you have to be careful about that. So that is what another principle of nutrigenomics, to find that which kind of diet will put an individual on higher risk of acquiring certain diseases. Okay, chronic disease onset. For example, examples of chronic diseases are all your cardiovascular diseases, liver disorders, kidney disorders. Okay, over a period of time. Okay, over a period of unhealthy lifestyle and unhealthy eating habits, which got accumulated, and that came up as a chronic disease. Okay, so to regulate the diet controlled genes, okay, and to prevent the chronic disease onset earlier, we focus on nutrigenomics. Okay, how an individual's body is going to react to a food. It is very subjective. Okay. Then your genetic makeup. The influence of diet on health and disease is likely to be affected by your genetic makeup. Okay, two kids from the same family, both of them are having Coca-Cola, chips, or something junk food. Okay, one child may not get the effect of it. The other child may get sensitivity reaction, bloating, diarrhea, constipation, something. Okay, why? Because both the kids have a different genetic makeup. Your genetics will decide how you react and respond to certain food. Then we have nutritional intervention. Nutritional intervention is from the viewpoint of nutrigenomics. Nutritional intervention will be given by the genetic clinics. Okay, clinics who are concerned with genetic counseling, etc. Okay, they give the nutritional intervention as well. So based on an individual's nutritional status, their genotype, phenotype, etc., their current health status, okay, what kind of food they should be having as a family, what kind of food they should be having, it will be noted down and it will be given to them as a part of their genetic counseling. Okay. Next, coming to the interactions between nutrition and gene. Okay, how can a nutrition, vitamin A, your vitamins, your minerals, okay, your macronutrient, etc. How do they affect your genes? Okay, so first is their direct interaction. So in this image, you can see. So this is a DNA protein, okay, which is an activator. And this is the binding site of the activator. This green thing is an activator. This is a nutrition. This is a nutrient. Okay, this activator. Let's consider this activator as a nutrient. Okay, and they come on the DNA where it has a binding site. Okay, the your nutrients, whatever you have, cannot just go anywhere on the DNA and cause some changes. Okay, there are specific sites on the DNA which is ready to accept a reaction from a nutrient. Okay, so once the activator is bound to the DNA, okay, a transcription will take place. Transcription means a change. Okay, a change will take place. This change can be positive or negative. Okay, if it is a negative, we call it as a mutation. Okay, so a transcription will take place, and a target gene, because DNA is made up of different forms of different sequences of genes. Okay, so as soon as the transcription will take place, a target gene will be affected. Okay, either it will be destroyed or it may change into something else. Okay, and it will show new signs and symptoms, which was shown all these years will cease to exist, and new signs and symptoms will be shown when there is a genetic change. Okay, so this is how a direct interaction can take place. And when you have a repressor, repressor is also your antibodies. Okay, certain antibodies are there in your body which wants to protect you from this unwanted mutation. Okay, your body has certain antibodies which will protect you against unwanted mutation, because as soon as an activator or a nutrient is always going to cause some change in your DNA, definitely you will not lead a healthy life. You will keep on changing. The way how your body responds will be changing, and you can't follow a routine, a sequence, or something like that. So naturally we have a repressor. Let's consider the repressor, this red circle. Okay, this red circle as an antibody. The repressor will cover up the area which is ready to accept a nutrient. Because of the presence of repressor, as soon as the activator comes here, it is not able to attach to the DNA. And when the activator cannot attach to the DNA, no change, no transcription, or no change will take place. Okay, no transcription will take place, no gene mutation will take place. So the person can continue a healthy life. Okay, so this is a direct interaction of the nutrition on your genes. Okay, if it is directly interacting, this is the way it will your nutrition can directly go and bind itself. Okay, your nutrient can directly go and bind itself to the DNA and cause some changes. But if there is a repressor could be your antibodies as well. Okay, if the repressor is already present. And some food, okay, like garlic and all, they are also natural repressors of genetic mutations. Okay, there are certain food, we will come to that later, certain food that are natural repressors of genetic mutation. They will not allow too much of changes, transcriptional changes to take place in the DNA. Okay, so this is the direct interaction. Is it clear to all? Direct interaction. So this is the DNA. Okay, DNA has an area where it is ready to accept an activator, and this activator can be any of your nutrient, all your vitamins, your minerals, your macronutrient, anything, vitamin like substances, etc. Okay, they all have a capacity to go and attach themselves, the metabolites of these, obviously the metabolites of all these nutrients. Okay, they have a capacity to go to specific areas of your DNA, attach themselves to your DNA. Okay, and once the nutrients get attached to your DNA, they possess the ability to cause some change. The change is called transcription, and a gene is targeted. A gene can undergo some changes. This change can be positive or negative. Negative is it can lead to some cancer like diseases, some chronic diseases. Okay, so that is how nutrients have a direct interaction with your DNA. This is how nutrient can have a direct interaction with DNA. Okay, but when the antibodies are present, this antibodies will cover up the activating the binding site. Okay, the antibodies will cover up the binding site in the DNA, so that when the metabolites of the nutrients come and try to attach themselves to the DNA, this area is not available. Okay, so there will be no transcription that will take place, and the target gene is safe from any kind of mutation. So this is a direct interaction of your nutrients which you eat and its effect on your genetics. All your antioxidant rich food, comes under the repressor category. Okay, we'll see that topic is coming up. Okay, all your antioxidant rich food will come under repressor category, because your free radicals, remember, not just the nutrients that have undergone metabolism, and out of that metabolism, free radicals are also present as a byproduct. These free radicals have enough capacity to cause this transcription. Okay, all your antioxidant rich food comes under the repressor category, which will not allow the transcription, unwanted transcription, to take place. Okay, then we have epigenetic. Epigenetic is indirect. Okay, kind of an indirect way of interaction. So your surrounding, how you behave in your environment, the environment where you live in. Okay, urban, rural. Okay, the kind of lifestyle you have, is it a fast-paced lifestyle or a slow, stress-free lifestyle? Okay, all these things have an effect on your DNA. Okay, all these things have an effect. Your environment has an effect on your DNA, based on which your nutrients will react. Okay, if you live in a stressful environment, okay, an urban, one tier city, etc. Okay, so when you reside in such cities, such metro cities, where the life is fast paced, okay, you do not get enough time for yourself to exercise, to de-stress yourself, etc. Okay, definitely free radicals are hitting on your DNA. Your DNA is undergoing some mutation, and when these mutations are taking place at the nutritional level, the way how your body is supposed to respond to metabolism, okay, the way how your body is supposed to metabolize, absorb the absorb the food, okay, definitely your nutritional health will also be impacted. Okay, so that is epigenetic and metabolism. Your external stress factors can impact your metabolism, okay, by changing your DNA structure. How does epigenetics? Epigenetics means everything outside your body, everything external to you. Okay, everything external to you. How is it changing your metabolism? The first answer is stress. Okay, stress can change the way how your body responds. Stress has already caused some mutation in your DNA, because of which the food which you earlier used to enjoy, which was easier for you to when you were in your early 20s and all. Okay, it was easier for you to have spicy food and sleep at night. Okay, but as soon as you grow near 30s, etc., you have something spicy or heavy at night, you will and when you go to sleep, you feel acidity, etc. Okay, so that is the way how stress has changed the way how your body works. Okay, internally, by changing the way how your DNA responds to various kind of nutrients. Okay, so that is epigenetic. Epigenetic is an indirect, it is not directly causing some change in the DNA. Okay, it is causing changes in your body like stress, okay, ulcers, some diseases, okay, uh, fatigue. These things, these are the clinical signs and symptoms, and over a period of time, when you accumulate stress, fatigue, etc., definitely because of these mental health conditions, your DNA will also undergo certain uh, wear and tear, and because of which it is going to respond to nutrients differently. Okay. Then we have genetic variation. Common genetic variation can modify genes expression or function. For example, um, a person who does not have any family history of cancer, okay, but out of the sudden they got infected by a cancerous gene, or something like that. They may have led a healthy lifestyle, etc., but for idiopathic reasons, for unknown reasons, genetic variation can take place. That is common genetic variation. Okay, sometimes it could be your hormonal imbalances, which does not have a proper cause or root cause which we can pinpoint to. Okay, because of hormonal imbalances, there was a genetic variation because of which it led to cancer or any benign tumors. Okay, so that is genetic variation. It is not affected by anything internal or anything external. Okay, there are no internal villains or external villains that have led to some genetic variation because of simple body processes. Okay, sometimes the DNA can overreact or underreact, and it causes some variation. Okay. Next, coming to the tools that we use in nutrigenomics. So genotype and phenotype. From people who are from the science background, can you like genotype, we know your genome, the genetic structure, etc. Okay, what we find in the nucleus, the genome, when you take that out and try to study it, we get the genotype. Okay, what kind of repetition, what kind of sequences this individual has, we can find it out. Okay, what is phenotype? Any examples can you share from real life? Yeah, external experience, appearances. Yeah, if a person is tall or short, these things becomes their phenotype. Okay, color of their skin, eye color, hair type, curly hair, straight hair, wavy hair. Okay, so these things are phenotype. Okay, each of these phenotype has a specific gene sequence in them. That is the genotype. Okay, so genotype is just the sequence. Okay, it is very technical. It is your nitrogenous sugar and the different sequences of genes which is found in the DNA. That is genotype. But how it manifests physically, that becomes the phenotype. Okay. So epigenomics, which we have discussed recently, whatever takes place outside your body, external, okay, some kind of changes it will undergo in your DNA. Okay, a genome has more than 25,000 genes, around 6.4 billion bases. Bases and genes are different. Okay, Bases are the C, T, A, G, which we have seen. Those are the bases. Okay. So through the DNA, we get the gene expression, okay, from RNA to protein and to metabolite. Metabolite are the ones which is the broken down or post metabolism, whatever is remaining, that is a metabolite. Okay. So this entire area, how the RNA registers protein, okay, how the protein after registering into RNA, what is remaining, okay, what is remaining, because protein has to break down, give out the nitrogen. Protein is the only macronutrient which has nitrogen, okay, nitrogen molecule in it. Okay, so the protein has to break down, it has to give up nitrogen for the repair and for the maintenance of the DNA and RNA. Okay, so after giving up nitrogen, what is remaining? Metabolite will be remaining. Okay, so these all function from RNA till you get the metabolite becomes the metagenomics. Okay, so transcriptionomics is the way how RNA changes. Okay, RNA uses certain enzymes. Okay, RNA uses certain enzymes to change a macronutrient into something else, to change protein into its metabolite. Okay, so the study of all these transcription enzymes is what we call as transcriptomics. Okay, the study of transcribing enzymes is what we call as transcriptomics. Okay, the way how proteins give up nitrogen molecule for the DNA maintenance, repair, etc. Okay, that is proteomics. Okay, and then the remaining metabolite. Okay, how is it discarded from the body? Because if metabolites stay in the area for pretty long time, they can cause further mutation. Okay, they have to be discarded, otherwise they may become toxic for the DNA. Okay, so studying the different kind of metabolite formed in the process of breaking down of protein, okay, that is called metabolomics. Okay, so I hope this is clear for you, what is the importance of protein here. Okay, and how metabolite can interfere with your DNA health, and what is the main role of RNA. The main role of RNA is to change something. Okay, it's to change something. To here we see how RNA is helping the protein to change itself into a metabolite after giving up the nitrogen molecule. So the study of all these transcriptomics, proteinomics, metabolomics, okay, genomic, these are the tools which we use in nutrigenomics. Okay, those who are from the science background, who have a research affinity also into genetics, they go for this clinical genetic counseling clinics, etc. They get work in genetic counseling clinics. Okay, so they also have to give some insight on what kind of nutrition, especially a couple who is planning to have a baby, okay, and a couple who has given birth to a child who has some genetic deformity. Okay, so in that case, what kind of diet should be given to the child? Okay, what kind of diet the parents have to take care of if they're planning future kids, etc. These kind of nutritional guidance counseling is given in genetic clinics as well. Okay, so for those nutritionists and dietitians who should be specifically from a science background. Okay, because if you have done biotechnology and all such courses, you will understand this much better. Okay, so is it clear what kind of tools we use? The names of this transcriptomics, proteinomics, metabolomics, okay, genomics, these are the tools which we use in nutrigenomics. Okay, so the meaning of all these studies have been explained. Okay, in a single statement, they have explained on page number 481. Just read that again. Okay, you just have to read those 4 lines in detail. They have given on page number 482, 483, etc. That those who are from science background, they can definitely read it. But from exam point of view, it's not important. If you want more knowledge about each one of these different type of studies, you can read these page numbers 482, 483, and 484. Okay, if you want some in-depth knowledge about this, you can read it. Otherwise, from exam point of view, just understand what is the meaning of each of these studies, and these are the tools which we use in nutrigenomics. Only that you have to keep it in mind. Okay, is it clear to all? So next coming to bioactive components of food. So how food acts at cellular level, how nutrients acts at cellular level. Okay, so first is your cell growth. So for your cell to cell usually cell will not grow per se. Okay, cells, all the cells in your body, they undergo proliferation and differentiation. Okay, people who are from the medical background, can you say something about these two terms, proliferation and differentiation? People from the medical background, what do you mean by cell proliferation and differentiation? Especially when it comes to the topic of cancer, these two words are very important. If you see the image here, if you see the image on this top left corner, you can see a cell proliferation taking place. A single cell, okay, in which this cell has a dark blue color, a green cell, okay, cell membrane, etc., and a dark blue nucleus. Okay, over a period of time, you can see the nucleus undergoes changes. It divides into two cells. A single cell, over a period of time, a nucleus undergoes changes. Okay, nucleus will divide it into two parts, two halves, along with which the cell membrane will also divide into two halves. Okay, so this is proliferation. From 1, it became 2. From 2, it will become 4. From 4, it will become 8, then 16, and so forth, so on. Okay, so that is proliferation. So for cell growth, cell proliferation, and differentiation, you require food. Okay, nutritious food. Apoptosis. Have you ever heard of this term, apoptosis? Can anyone guess what will this mean? Yeah, apoptosis means cell death. Okay, cell death. Cell death has to take place. Cell death has to take place. Otherwise, if a cell stays on for a pretty long time, it can undergo some genetic mutation. It can lead to some tumors or some malignant tumors, that is cancer. Okay, so after a period of time, every cell has to undergo apoptosis. Okay, either undergo proliferation and then undergo apoptosis. Okay, that is cell death, so that new and replenished cells can take over. Okay, so there are certain food components. We'll come to that later. Some certain food components like garlic, some antioxidants. Okay, the cell which has undergone certain kind of mutation. Okay, as a defense response of our body, as our body's defense response, there are already some antibodies present that will be triggered by the cell mutation. It will go and destroy that cell. Okay, and that is also called apoptosis. Certain food components, certain antioxidant rich food can also do the same thing what our antibodies do. Okay, these nutritional food can also identify the cells that are undergoing some mutation, or the cells that are at the risk of some certain mutation, and it will kill that cells then and there itself to protect the human body from cancer. Okay, most of the cells will undergo proliferation and differentiation, and some will undergo apoptosis, that is cell death. Then repairing the DNA, protein-rich food. All your protein-rich food and antioxidant-rich food will do this job of repairing the DNA. Okay. Inflammatory response, to reduce the inflammatory response. And there are some anti-inflammatory food. Okay. Anti-inflammatory food, anything that is vitamin C rich food. Okay. Vitamin C rich food are anti-inflammatory. Okay. Hormonal regulation is also done by your food nutrition. Cell differentiation, the breakdown of cells into and the increase of cells. Okay, that is cell differentiation. Carcinogenesis. So this is you can see here. It's a microscopic way of how a cell can be represented. The blue area you can see, this is a tumorous zone. Okay, this is the cell which does not have a clear differentiation. Okay, a healthy cell, healthy tissue will have cells that have a pretty clear differentiation, which you can see on the either side of this particular pic. Okay, on either side of the photo you can see clear differentiation between the cells. Okay, there are a separate demarcation of line, everything is clear. Okay, where each and all cells are placed in a straight line and everything. Okay, almost straight line and everything, it is clear. But as soon as the cell undergoes mutation, okay, the cell proliferation and differentiation will get impacted. Okay, you can see here the blue bunch of cells. Okay, they do not have a clear line, they do not have a normal size as compared to its peer cells. Okay, the way how they proliferate and differentiate is not healthy, it's not normal. So that is carcinogenesis. This is how cancer starts. Genesis means start. This is how cancer starts. Certain food components can also lead to carcinogenesis. Okay. Yeah, turmeric is also known to have this anti-inflammatory response, as well as it can also scavenge out the cancer causing mutation cells. Okay. So the food components, which if you want to know exactly what food you should have, etc., please read page number 485 from 484, bioactive food components, and page number 485. You can put it in the chat box as well. Those who do not have the textbook, new students here, those who do not have the textbook, you can put some examples. Those who have the textbook, you can read and put some examples right now live in the chat box. It can help others as well. Turmeric, tea, grapes, chicory, which is found in filter coffee. Okay, chicory is not found in your instant coffees much, but if you are used to having South Indian filter coffee, etc., it is 70% your Arabica coffee and it is 30% chicory. Okay, so usually in the southern part of India, the coffee which you have naturally has chicory mixed into it. Okay. Yeah, fenugreek. Yeah, omega-3, omega-6 will also come under cancer preventing food. Green tea, why? Because it has the flavonoids, theaflavins, etc. Next, coming to how a chronic disorder, okay, how a chronic disorder is related to your genetics. Okay, so this pathophysiology goes up and down. Okay, so your diet, okay, a healthy diet or a bad diet, you can choose which form you want here. Okay, so earliest markers of food bioactivity is decided by your diet, the kind of diet which you're choosing. If you're choosing a healthy diet, a home cooked diet, antioxidant rich meals, etc., on a daily basis, okay, that will lead you to a positive food bioactivity. But if it is the opposite, if you have a lot of synthetic food in your on a regular basis, etc., Okay, junk food, synthetic food that stresses your body out. Okay, even if it gives you the pleasure of indulging, okay, but it stresses your body at a metabolic level. The food that can cause stress to your body, to your digestive system. Okay, that could lead to negative food bioactivity. Okay, bioactivity means at the cellular level, how the food, how the nutrient is going to act at a cellular level. Okay, so the diet, the earliest food markers, it will cause pivotal means it's a turning point. Okay, turning point of your gene expression. This is where the transcription has begun, the mutation has begun. Okay, you can call this as a mutation area also. Okay, and once the mutation has begun, definitely there will be an abnormal gene expression. Okay, after a mutation, a normal gene becomes the target of the mutation, and it becomes an abnormal gene. Okay, and cellular dysfunction, the way how a cell is supposed to normally function in that particular area, post mutation, it will not function the same way, because the cell, if when the cell undergoes undifferentiated proliferation. Okay, the way if you remember the previous image, I have shown you a bunch of cells. Okay, the pink cells were well differentiated, but the blue bunch of cells were not well differentiated. Okay, so the blue bunch of cells which underwent quick proliferation differentiation without any demarcations, etc., they are not functional. They do not function properly. Okay, they are not healthy cells. They are not considered healthy. They can't function properly. So definitely it leads to cellular dysfunction. Okay, and when at cellular level your cells are not supposed to do what it is supposed to do, that is when you acquire a disease. Okay, so this is the pathophysiology of how your diet can lead to disease. Okay, and this is one way of seeing things. It can go from the other way also. Let's say you acquired some gastric infection. Okay, let's say you have some peptic ulcer. Okay, peptic ulcer. I guess we do you know what is peptic ulcer? All of you, have you heard about this term, peptic ulcer? Stomach ulcers. Okay, ulceration in the stomach. Sometimes in the lower part of your esophagus also you can get ulcer. Okay, so peptic ulcer can be I guess you can imagine what can trigger peptic ulcer. Can you give us the students who are from the medical background, just quickly give what could lead to peptic ulcer from dietary point of view and lifestyle point of view? Spicy food, fried food, having too much of acidic food, citrusy food, stress, smoking. Yes, smoking, sedentary lifestyle, definitely. Fasting for prolonged fasting. Okay, prolonged fasting can lead to peptic ulcer. Keeping the stomach empty for a long time, and when the hydrochloric acid concentration. And one more main thing, not having enough water. You fast for a pretty long time and you do not give yourself, if you do not keep yourself hydrated, what happens? The concentration of the hydrochloric acid will increase. Okay, and that will lead to peptic ulcer. Too much of consumption of caffeinated drinks, like too much of coffees and tea. Okay, yeah, these all are the causative factors of peptic ulcer. Okay, so peptic ulcer will cause a cellular dysfunction because it has caused lesions. Okay, it has caused a wound. Ulcer means what? It is a wound. Ulcer is a wound. Okay, it's an internal wound which is bleeding. Peptic ulcers will bleed. Okay, because one of the signs and symptoms of peptic ulcer is when you pass the motion. A person who has peptic ulcer, if there was too much of bleeding from the ulcers, okay, when they pass the motion, there will be black spots in their feces. which means the blood clot is there. Blood clot is there means internal bleeding was there. Okay, so cellular dysfunction. Why the peptic ulcer led to cellular dysfunction? Because there was a lesion there. Wound healing was not done properly. Okay, a scar has been formed, because once a wound has been formed in your stomach, it will not go back to the normal way how it was before the ulcer. Okay, when it heals, there will be scar, just like on top of your skin. When you fall, when you get a cut or a bruise, okay, and when you heal, there is always a mark that is left behind, a scar that is left behind, and that area will not function as before. Okay, you will not sweat from your scars. If you have a big scar on your any part of your body, if you see, that area will not sweat, that area will not produce sebum, so the function is gone, cellular function is gone. Okay, and if the wound healing is not done, if the wound is still open, if you do not treat a peptic ulcer, the wound healing is not done, it is still open, it will cause mutation. Okay, because the wound, it is very difficult for such peptic ulcer to heal on its own, because it is in a place where it is acidic. Okay, the stomach is naturally acidic, and you have a wound there, you have an ulcer there. When your stomach moves, when your stomach tries to churn, contract and relax and try to churn the food, definitely the hydrochloric acid will attack the wound, and it will prevent the wound healing, it will delay the wound healing. So delayed wound healing will cause much more complications. Okay, so the complication over complication, genetic mutation will take place, abnormal gene expression will take place. Okay, and then you have to change your diet as well. And once you reach a point where your peptic ulcer is affecting your lifestyle, you will have to change your diet. You are no longer allowed to drink tea or coffee. Okay, you will not be allowed to have spicy food, you will be put on bland diet. Okay, and sometimes surgical interventions are also required in complicated cases of peptic ulcers. Okay, so your diet will change. So this is the pathophysiology. It runs both the ways, from diet to disease and from disease to diet, based on gene expression. Is it clear to all? Just focus on this central arrow. Okay, things that are written on the periphery is not that important. Okay, so coming to your diet and the risk of getting a cardiovascular disease. Okay, coronary heart disease or cardiovascular disease. Okay, CAD stands for cardiovascular disease, or cardiac or coronary artery disease. Both we can take in this condition. Okay, so people who have a low genetic risk, okay, family, they do not have a family history of any coronary artery disease. Okay, and when they follow a healthy lifestyle, this is the dissection of the arteries and veins, how they look like. Okay, when they follow a healthy lifestyle, there is no plaque that is accumulated in the arteries, so they will not get any coronary artery disease. But people who have a low genetic risk of heart diseases, but still they have a very poor lifestyle, they can also definitely, it can definitely lead to any form of heart diseases. Okay, not just CAD, CVD, or any form of heart diseases. Okay, people who have a high genetic risk of coronary artery diseases. Okay, even if they children can also develop peptic ulcer if the diet is not under control. From a very prolonged time, or from a very young age, if you are giving your child tea, coffee, too much of spicy food, definitely. Okay, because their stomach is in the growing phase. Kids, their stomach is in the growing phase, it is not that strong as compared to an adult. Okay, so children can also suffer from peptic ulcer. Rare, but it can happen. So people who have a high risk of coronary artery disease based on the family history, etc., when they follow healthy lifestyle, still they will be, based on the genetics, they will still have a natural inclination of accumulating plaque on their arteries, inside their arteries. Okay, and definitely if they have a poor lifestyle, definitely they will fall on the high risk zone. The disease will start. Okay. So just because you have a low genetic risk doesn't mean you can indulge in a poor lifestyle. Even with low genetic risk or high genetic risk, you have to maintain a healthy lifestyle to prevent any chronic diseases. That's all